many researchers have used the biologically oriented approach to find the location of the splice signals in the intron-exon junctions. So we can see here there exists a weakly conserved sequence of 8 nucleotides at the boundary of the exon and introns. So we can see in this diagram that there are 2 exons and 1 intron, and at the boundary of exon intron we have a conserved sequence of 8 nucleotides at the boundary of an exon and an intron, and we know intron is a donor splice site, and a sequence of 4 nucleotides at the boundary of an intron exon acceptor splice site. So at this site, that is the donor splice site, we have a total of 8 nucleotides, and at the boundary of acceptor splice site we have conserved nucleotides that are only 4 in number. And we can see also that below the nucleotides their frequencies are also mentioned. For example, the C cytosine has a frequency of 78%. A and G, adenine and guanine, because they are written in green, green means 100%, so they both have 100% frequency. Similarly, G have 55%, and we can see also at the donor splice site that all the frequencies of the nucleotides, they are written below their nucleotides. So these conserved regions we can detect, and we can predict the location of the intron and exon junction. So the profiles for the splice sites are weak, and these approaches have led limited success and have been supplemented by hidden Markov model that captures statistical dependencies between sites. So a popular example of this later approach is GenScan. This is a software which was developed in 1997 by Chris Burge and Samuel Carlin. So this GenScan combines coding region and the splicing signal predictions into the signal framework. So the splice site prediction is more reliable if we find the coding regions on one side of the site, and on the other side there should be no coding regions. So such statistics are being used in hidden Markov model frames of the GenScan, which combines the different statistics, for example the splice region statistics, the coding region statistics, and the conserved regions present near the start of the transcription, this apart from the other parameters. So the accuracy of the GenScan software is less when the exons present in the gene is less in size, or they use the unusual codons uses when they code for the amino acids.